Hello everybody, how are you? Hopefully you are doing well at home. As I promised, I am here with the structure of DNA. So, at first we will know the full form of DNA. Full form of DNA is deoxyribonucleic acid. So, D means deoxyribose sugar and Na means nucleic acid. Now, this is a structure of DNA. This is called double helical model of DNA. You can say the spiral, it is looking like a spiral staircase. Okay, like coiling rope. Okay, if you see the coiling rope, you can see how the ropes coiled. Okay, DNA is just like that. It's tightly coiled inside the chromosome. Okay, so that's why it can stay here, stay inside the chromosome, and such a long structured DNA, as I said, but it can get the space inside the chromosome, inside the nucleus. Okay. Now what happened in this spiral staircase? You can see how it is looking like a spiral body. Okay. Now what happened? The DNA is actually a polymer. Polymer means this is in DNA you can see DNA is a nucleic acid bond. So here in DNA, polymer means nucleic acid. Okay. So poly means so many. Many means many. And monomer, polymer means so many monomers joined together. Like if you see the structure of starch. Starch is made up of so many glucose joined together and make starch. Now what happened here also you can see the polymers means so many monomers joined together. And in case of DNA, polymer means nucleic acid. Okay. And here nucleic acid is made up of so many nucleotides. Nucleotides joined together and form nucleic acid. One DNA strand, the DNA strands, this is a strand and that is one strand. That is one strand, that is another strand. Okay. This DNA strand is made up of so many polynucleotides. Poly means many. Okay. Polynucleotides means so many nucleotides joined together and make this DNA. So this, when the polynucleotides join and make the DNA, it is the structure. The structure is called double helical model. The structure is called double helical model. It is given by Watson and Crick. Okay. And for that in the year 1962, they got Nobel Prize also. Okay. Now one important thing is nucleotide. Now what is nucleotide? Nucleotide is mainly made up of three things. Nitrogen, base, okay, then sugar and phosphate. So here I have shown a structure how they have joined. Suppose this is the phosphate and this is the sugar and this is the nitrogen base. The sugar is actually deoxyribose sugar. This is a 5-carbon sugar. Okay, 5-carbon sugar. Now come to this double helical structure, how it is joined here. You can see here. Up to this, it is clear I think. Now I am going to discuss about the details part. Here in this details part you can see in this structure you can see that we have learned that the middle part is made actually nitrogen base part. See the chains here. The chain is actually made by sugar and phosphate backbone. This is a backbone made by sugar and phosphate. But the middle part of the chain, this is one chain, this is another chain. The middle part of the chain is called base pair. Okay. So the base pair is actually made by nitrogen base. Okay, nitrogen base. And there are two types of nitrogen base are there. One is purine. Another one is pyrimidine. Pyrimidine. So, purine and pyrimidine is again, there are two types of purine. This is adenine and another one is guanine. Pyrimidine has two parts also, that is thymine and cytosine. So pyrimidine has two types, adenine A and guanine G. Thymine means T and cytosine means C. Remember, the number of purine and pyrimidine is equal in DNA. Okay, it means adenine plus guanine equal to thymine plus cytosine. Now the thing is that when they create the bond, what happened? There are weak hydrogen bonds are there to make this bond. Purine always make bond with pyrimidine. Okay, purine make bond with pyrimidine. Now what happened? Here you can see adenine always make a bond with thymine and guanine always make a bond with cytosine. And the bond is made by weak hydrogen. Okay, and here you can see adenine is make a bond with thymine with two hydrogen bond, and guanine join with cytosine with three hydrogen bonds. That is the difference between these two. Now what happened? You can see adenine and guanine. These two means purine has two ring structures. Okay, they have two rings. Two rings. This is guanine also and adenine also. But the pyrimidine, thymine and cytosine has what? One ring only. They have only one ring. Remember, one easy thing I want to say you. Pyrimidine, you can remember pyrimidine. Suppose pyrimidine means we have learned that thymine and cytosine. So, how can it is a very easy way to remember which one is pyrimidine. See, in pyrimidine, Y is there. In thymine also, Y is there. Cytosine, Y is there. So, you can remember easily which one is pyrimidine and which one is purine. If it is Y is there, pyrimidine spelling, Y is there. Thymine spelling also Y is there. Cytosine spelling also Y is there. So, it is easy way to remember. Okay. Next, come to the point. We can see the gap or distance between. See, this is the double helical model of DNA. Okay, it's a spiral staircase looking like. The one coil and one turn. Okay, in this one turn, the distance is 34 angstrom. Okay, or 3.4 nanometer. It is a microscopical unit which we can use. Whatever thing we can see under microscope, we use this unit of measurement. So, this one coil, distance between two coils, here to here, the distance is 34 angstrom or 3.4 nanometer. If you come to the width is 20 angstrom, here 20 angstrom or 2 nanometer. Okay. If you comes to the gap between two nitrogen bases, this is one nitrogen base, next one is another nitrogen base. So, in between these two nitrogen base, the gap is 3.4 angstrom or 0.34 nanometer. Okay. And one distance between two coils, we get 10 nitrogen base pairs. Okay. This is one nitrogen base pair, this is another nitrogen base pair, one nitrogen base pair, another nitrogen base pair. Like this way, 10 nitrogen base pairs are present there. Now, we'll go to more detail. What it is there? We have learned there. The chain is made by phosphate and sugar. Deoxyribose sugar. Okay. So here you can see this one is sugar, and these are nitrogen base. This is also nitrogen base. We can give the name also. As I said that guanine and adenine has 2 rings. Okay, in guanine you can see 2 rings are there, but here it is thymine. Thymine has 1 ring only. This is one nucleotide and this is another nucleotide. These nucleotides are joined with the phosphate. Here 2 phosphate you see from here 1 phosphate joined with the deoxyribose sugar, and here is the nitrogen base. In another part also we can see another nitrogen base is there. So in this case we can see how it is joined. This is a 5 carbon sugar. Okay, if we go to clockwise 1, 2, 3, 4. This is another bond. You can see the 3rd carbon hand is joined with the phosphate, and in this case 1 phosphate group actually joined with 2 sugar. 2 sugar carbon means one is 3rd, another one is 5th. In above part is joined with 3rd carbon. In down part it joined with what? 5th carbon. Everywhere it is like that, and this way this make the bond. Nucleotides after nucleotides after nucleotides, and this is only one part of the chain. Same way in that part the bond will again join and make the total chain. Okay, so this part is only half part you can take. Okay, so guanine, adenine will make the bond with thymine and guanine and cytosine. So guanine will make a bond with cytosine, and thymine will make a bond with adenine. Like this way the bond will make. So we understood this one. Here if it is 2 hydrogen bond, here it is 3 hydrogen bond, like this way. This is a complete structure of a double helical model of DNA. Now let's discuss about gene. Gene is nothing but a part of DNA. Gene. Before that, we have to know what is the function of DNA. We know that DNA present inside the nucleus is tightly coiled in the chromosome. Now what happens? What they do? They contain the genetic code and they passes information from parents to the offspring. Now which part actually you want? Gene is actually the part of a DNA. Any part of a DNA. Suppose this part. This is a gene. The portion of the DNA. And they present in linear position. Straight one after another like this one. And the gene actually they present and take different genes used for different functions, and they present according to like that way. Okay. And one time only one set of gene can perform one function. Suppose if I take that this part of gene, DNA, this part is actually one gene for responsible for one person's red hair. Suppose one person has red hair, okay, and blue eyes. So this portion of DNA is called the gene which carries the red hair character, and this portion of DNA carries the characteristic of blue eyes. This is the one gene, this is another gene. So, short portion of DNA. and every gene has specific position and function. So it is always a confusing matter. Which one is gene, which one is DNA? Now it is clear that gene is that nothing but a portion of DNA which carries different character, and for that they present in different position. Understood? I think it is very clear now. So today up to this, the structure of DNA, structure of gene. So in my next class I am going to start tissue. Okay. So this is a continuation, and all the link of cell you will get my description box. Okay, one more thing here I want to say, that the portion when I discussed nucleus, I already discussed a little bit about DNA, how it is present in chromosome. And one thing, remember in ICSE syllabus it is not there, but you can follow my cell division part, where the genetic exchange happen in meiosis 1. So this way the gene change from parents, some genes part come from father, some genes come from mother, and they form a new chromosome. So this way the portion of genes come from parents to offspring. For that you have to follow my meiosis cell division part, and you can see this one in meiosis 1 I have discussed, right, this property. Okay. So up to this, and if you like it, you give a like and you share it. How much you share it, this much I can reach to the students. Okay, and don't forget to subscribe my channel. Okay, thank you.